I'm Christer Ericsson. I'm a research scientist at the Karolinska Institute, and I'm also the Chief Scientific Officer of Icellate Medical, which is our new name, previously liquid biopsy. So we have developed a new way to ultimately diagnose metastatic cancer by actually capturing those cells that mediate the metastases from blood samples. And this is predicated on the well-established knowledge. Oh, doesn't seem to. Oh, here we go. That it's the metastases that are associated with the majority of deaths associated with cancer, not the primary tumor normally. And it's been established fairly recently that in a number of cases, at least, the metastases are spread early in the disease, not as was previously thought very late. They're spread through the blood, and it's a few individual cells that ultimately nucleate those metastases that may in the end kill the patient. So those, it's been very hard to isolate those cells. We've known about the likely existence of these cells for about 150 years, but only in recent time have we been able to do something about it. And so one of the reasons is they're very rare, individual cells per milliliter of blood or less. Let's see. So not only are they rare, they're also different from each other, and they evolve. They're plastic. So this makes it like looking through the proverbial haystack for a needle, but in this case a needle which doesn't always necessarily look like a needle, but is also changing. So it's understandable that this is a quite difficult thing. In this process of change, they become difficult to detect, but it's also part of what makes them so harmful to us. So it's well known that cancers are evolved because of rampant mutations, but these rampant mutations also, at least in many cases, set off, it is believed, a program called epithelial to mesenchymal transition, which changes the appearance of those cells from being fundamentally a kind of cell that's termed epithelial to something more mesenchymal, and in the process changing several fundamental and important properties of those cells. So for example, losing cell adhesion molecules, so they sit loose and they are more likely to be able to migrate. Also to acquire migratory or invasive properties, and also gaining stem cell properties. Now, so this EMT program and others will change the properties of cells to make them more dangerous, but also more difficult to isolate. And this is because the existing technologies available today that use antibodies to fish out those cells from blood samples are based on the assumption that the cells are fundamentally epithelial still. And so what's used mostly is this particular molecule here, which is cell surface molecule called epithelial cell adhesion molecule, which however is often lost. And so when it is lost, those circulating tumor cells can no longer be isolated with the help of that antibody. And that's real bad, because it looks like that group of cells that lose the marker may be some of the most malignant ones. So there's an urgent and well-known need in the field to be able to isolate also the EpCAM negative cells. And so that's where we've directed our efforts. And just to show you what this might look like in practice, we're looking. Oops, sorry. Here we go. If you focus on this center lane here, this is a number of different breast cancer cell lines. It doesn't really matter what they are, but some of them are fairly epithelial still in character, and that's shown by this green labeling of the EpCAM molecule on the cell surface. And this particular cell line is looks morphologically normal, almost. While the next one is more disorganized and has a lower expression level of EpCAM. And then you can continue down the line like that. And these two cell lines down here, which are very invasive. They have, in fact, from all practical points of view, lost the EpCAM marker. So current technologies to isolate circulating tumor cells is based on recognizing this, while the more invasive ones actually lack it. So this is what current technology can do. This is what we can do, and I'll show you that. So this is the prototype instrument as it looks right now. This is what we call the prototype 2, P2. We're now at prototype 4 stage. What this does is it has about the 10-minute processing times, and in practical terms, what you do is you put a test tube with a regular blood sample right here on this input port here. A regular sample consisting of about 7.5 milliliters of blood. It gets suctioned into this central processing unit, and you can look at it as it goes through because this is see-through. And then we go through about 4 iterative steps, or as many as we need, to get the pure sample. And you can see it happening before your very eyes. So you see the red color from the erythrocytes being sorted to one side and the clear circulating tumor cells to the other. And here's the results after our processing. And what we've done in this case is to isolate EpCAM positive and EpCAM negative cells spiked into a healthy blood sample, calculated the numbers we added, and counted how many we got out. And in this case, the blue ones you see here are the EpCAM positive ones that current day technology would be able to pick up, and the one with the label red nuclei are the ones that current day technologies would be blind to. So we recover both of those, and we have a high recovery yield. And the high recovery yield is important because there are so few of these cells. So to get any kind of statistics, you wanna isolate as many as you can of those. Now, what about patients? We actually have a case study here of a patient suffering from a metastatic colorectal cancer. And we were able to get blood samples from this patient, and you now see in large magnification here a circulating tumor cell isolated from the blood of this patient. So the blue here is the cell nucleus, the red is the cytoplasm. And this is cells isolated with the current definition of circulating tumor cells, which is a nucleated cell with an epithelial style intermediate filaments and not an immune cell based on surface markers. And so in this case, we could see there was a fairly large number of cells in the blood of this person, about 5 per milliliter. Immediately after treatment, heroic surgery going on for about 18 hours, there was a small reduction, and after about 1 month, there were no detectable circulating tumor cells anymore, down to the level of negative control healthy relative of the patients. So we would have predicted, based on this case of one, that he would have a better prognosis than normal, and it turned out he did. And this is now 3 years ago, and he's still well enough to work part-time. All right, so critical thinkers will ask, well, are those cells, how can we know those cells are actually cancer cells? And this is our current answer to this. Those cells that you saw on the previous slide were actually subjected to fluorescent in situ hybridization technology to look at amplifications or deletions in the genome for some loci, three loci in this case. And the big red and yellow blotches here are these amplified regions of the cyclin E and the c-Myc loci. So this would indicate that these cells are actually genetically aberrant and so most likely cancer cells. So we're probably, in all likelihood, isolating the right guys. So this shows that we can isolate these cells irrespective of surface markers, so it would be a universal method, and we can also subject them to meaningful downstream analyses, which is also very important. And so this little project has been seeing some interest, and we're we're getting some nice funding and collaboration from Roche in studying basic performance parameters and benchmarking, and we really appreciate the input. But we've already going on and exploring the potential of this technology to use it for cancer screening. Like I mentioned, metastases are spread early in the disease, it seems, and so it's quite possible people walk around apparently healthy, but still are in the early stages of suffering from metastatic disease. And so we've just initiated this study in South America, and we're screening blood samples from a initial test population of about 10,000 people at this point. And we have some initial positives from that, and it remains to be seen what the outcome of that will be. So that's pretty much the current status. So in summary, we have a technology which may well provide samples for just about everything to do with cancer, from early detection, estimating prognosis, predicting best treatment, and following the efficacy of treatment, as you saw, to potentially predicting new therapy targets, can be used in clinical trials or for basic research. So with that, I would like to thank some of the people who have been involved in the project and some of the funding so far. So we're funded by grant money at this point, completely. So with that, I thank you for your attention. I'll be happy to take some questions.